Hello, my name is Sammy Barmada. I'm a neurologist in the Cognitive Disorders Division here at University of Michigan. I'm also the director of the Brain Bank, and I run a research program that centers on disease mechanisms in amyotrophic lateral sclerosis and frontotemporal dementia. And so today, I'm really going to be going over these two conditions, looking at their underlying genetics and pathophysiology and how they may be related to one another. Very quickly, my disclosures, nothing relevant to today's presentation. I do have the pleasure of working with many different individuals at biotech and industry. All right, so just some of the objectives I want to go over today. First, I'm going to talk about how and why ALS and FTD may be related to one another. I'll then spend a little bit of time going into genetics and the pathophysiology, and at the conclusion, just highlight some directions, new directions the research is taking, highlighting biomarkers for both conditions as well as potential therapies. All right, so to start off with, I'm really going to talk about the clinical spectrum that we've observed between ALS and FTD, and then go into several specific topics as listed here. So when we think of amyotrophic lateral sclerosis, or ALS, many people think about this man. This is Lou Gehrig, incredible athlete, baseball player in the 1930s, who contracted this illness when he was 36 years old and passed away only a few years later. Really demonstrating how relentless it can be. Those of us in neurology also think about this man. This is Jean-Martin Charcot, who was a neurologist who actually did some of the first work on anatomical neuropathology, trying to relate symptoms of disease to what we see on pathology. And so what Charcot noticed was that people with progressive weakness really fell into two different camps. There were those that were weak, but their muscles were rather stiff. And there were those who were weak and they had a shrinkage of their muscles and they were kind of doughy. And he noticed that on post-mortem examination that those with that sort of weak and stiff pattern had dropout of the motor neurons that extend from the brain to the spinal cord, whereas those that had the muscle shrinkage and the weakness had a degeneration of the motor neurons that go from the spinal cord to the muscle, the lower motor neuron. And we can see this actually in the cross section of the spinal cord. We know that the upper motor neurons, the axons of the upper motor neurons, travel in the lateral aspects of the cord. This is the lateral corticospinal tract. And what Charcot noticed is that this section of the cord became scarred, and that's where the lateral sclerosis came from. On the other hand, the people who had lower motor neuron pattern with a muscle shrinkage had dropout of the cell bodies of the lower motor neurons that are present in the anterior horn of the spinal cord. This is where the atrophy, or the amyotrophic component of the name, comes from. And to this day, ALS is one of the few conditions that demonstrates both upper and lower motor neuron disease, and that's the origin of the amyotrophic lateral sclerosis, or ALS, that we use to this day. So as neurologists, we have to have more specific words for these things. So instead of the stiffness, we use spasticity, which is a velocity-dependent change in tone. So when the muscle is moved very quickly, there's a catch. Hyperreflexia, so when you lose the inhibitory tone from the upper motor neuron, reflexes become disinhibited, as opposed to lower motor neuron phenotypes where you have very low tone, that's the doughiness, and then absence of reflexes, or hyporeflexia. And so whenever you see a mix of these findings in the same person in different muscle groups, ALS really goes to the top of the list. So this is what, traditionally in neurology, this is the basis for clinical diagnosis, right? We do an examination, you see these findings, we can localize these findings to different aspects of the nervous system, in this case, upper and lower motor neurons, and we know that ALS, as I mentioned before, is one of the few conditions that affects both upper and lower motor neurons. And now we know that despite the large variation in the presentation of ALS, over 95% of people with this disease have a similar molecular neuropathology that's characterized by the cytoplasmic accumulation of this otherwise nuclear RNA binding protein called TDP43, which we'll talk about a lot. But the interesting thing is that there are other conditions that appear very distinct on a clinical basis, yet share the same neuropathology. And so, for instance, people who have personality change, word finding difficulties, and maybe avolition or a lack of motivation, we would localize this, generally speaking, to the frontal and temporal lobes. And based on the specific pattern, we might call this frontotemporal dementia. And yet over half of these people also have TDP43 cytoplasmic accumulation and nuclear exclusion, really calling into question our sort of, our clinical differentiation of these disorders. If they have the same pathology, yet they affect different parts of the nervous system, they're going to have different clinical presentations, but are they different diseases? This has a lot of parallels, I think, to stroke, in which people can present with any number of distinct clinical presentations, and yet we still call that one disease. And yet here we have one neuropathology showing up in different ways, but we call it more than one disease. So just a little bit more, if these two diseases are actually related to one another, then why don't we see more overlap in the presentation? Well, actually you do. So it just depends on how hard you look. So if you do very detailed neuropsychological testing in people, very detailed neurologic examination in people with frontotemporal dementia, you see up to a third have signs of ALS. On the other hand, if you do the neuropsychological testing in people with ALS, up to half will show signs of frontotemporal dementia. And this is really the basis for that spectrum that I mentioned before, where there may be a limited number of pathologies, including TDP43. If those affect primarily motor neurons, you end up with a clinical disorder that we label as ALS, whereas if they affect certain population of neurons in the frontal and temporal cortices, you end up with a condition that we label frontotemporal dementia. So what are those cell types? I think many neurodegenerative conditions, there is a one or more particular subtype of neurons that are affected or selectively vulnerable in these conditions that gives rise to the unique clinical presentations. But what are they in ALS and FTD? For instance, in Huntington's disease, we think of medium spiny neurons in the basal ganglia. Alzheimer's disease, there are many cell types that are affected, but one particular example is large pyramidal neurons in the medial temporal lobes. And in Parkinson's disease, the dopaminergic neurons of the substantia nigra pars compacta. Right, so this is, all kind of go along with the clinical presentations of these disorders, whether or not they're movement or cognitive in nature. But what about in ALS and FTD? Well, as we just talked about, in ALS, the primary cell type that is affected is the motor neuron, whether that's the upper motor neuron in the motor strip in the cortex or the lower motor neuron in the anterior horn of the spinal cord. FTD is a little bit different, though. So the one question is there even a selectively vulnerable neuron subtype in FTD that is responsible for the unique symptoms that we think about with FTD, including personality change, behavior change, language disorders? Well, so it turns out that the answer is maybe. So there are a type of long projection neuron that connect different association cortices called the von Economo neurons, or VENs. These are really fascinating cells that seem to be concentrated in the anterior cingulate cortex, which is one of the areas that is initially and primarily affected in FTD. These sort of bipolar cells that have very long axons and a single long dendrite, they tend to be much more common or only present in animals that have what I would say complex social structures. So that's a vague term, but what I will say is they're not present in every animal in which we look at. So for example, they're not present in mice, and they're not present in rats, which makes them very hard to study because those happen to be two of the species that we work on most often, but they are present in non-human primates, walruses, zebra, manatees. I don't know who decided to look at this in a pygmy hippopotamus, but they're there as well. And if you look at humans compared to, say, non-human primates, they're substantially more numerous, and even in the as we develop over the first few years, they become much more prominent. So the interesting thing is when we go back and try to look in people with frontotemporal dementia, these cells seem to be preferentially affected, and they are not affected in other neurological conditions like Alzheimer's disease. And this gives rise to the hypothesis, at least, that von Economo neurons in a related cell called a fork cell, because they have two primary dendrites rather than one, are the substrate of FTD. I think that's still, still to be seen. The biggest problem here is that they're very difficult to study, as I mentioned, and not only do you need the model system in which these cells exist, also we don't know a good way of distinguishing these cells other than their morphology, which makes their creation in the laboratory, say from induced pluripotent stem cells, very challenging. All right, so what about the epidemiology of ALS and FTD? So just in general, I'll start with ALS, but I think the numbers are largely similar with frontotemporal dementia. The incidence is about 3 per 100,000, which means every year among a population of 100,000 people, 3 will be diagnosed with ALS. So if you think about Ann Arbor, that's about 3 people. But larger cities like San Francisco, you end up with more, like 8 times more, and then New York, you have a lot. So it's not small, but the problem is that the death rate is so high. So in 3 years, half of those people will have died, and in 5 years, 3/4 will have died. So even though the incidence of ALS is about the same as something like multiple sclerosis, the prevalence is low because it's such a lethal condition, and a similar argument can be made for frontotemporal dementia. And that's why we think of this as a rare disease. FTD, there's an added complication in that it's really hard to diagnose, and I would argue that outside of tertiary care facilities, it is underdiagnosed. So in terms of the age at which these conditions present, so ALS, the peak presentation is actually in the early 70s. And again, I think, when we hear about ALS, we most often hear about it in young athletes, and I think that's because these are not the most common ones, but they tend to gain the most attention. FTD, the peak is a little bit earlier, in the early to mid 60s, and as a matter of fact, frontotemporal dementia is just as common, if not more common, than Alzheimer's disease in those 65 or younger. So what about inheritance pattern? So ALS, if you were to see 10 people in the clinic with ALS, 9 out of those 10 people have no substantial family history, whereas 1 out of 10 have a family history of ALS. Other things that we know are associated with higher risk of developing ALS include being a male, unfortunately, for some of us, So about a 1.3-fold increase in the risk. Military service, in particular active military service, and using firearms. Traumatic injury, especially repetitive, non-concussive injury to the head, and pesticide, chemical exposures. FTD, it's a little bit different. We think that there's a larger familial component to FTD. Again, it can be very challenging to figure out what is a positive versus a negative family history. But again, being a guy is unfortunately a risk. All right. So that sort of feeds into the next topic, and that's genetics. And so if these conditions are truly located on a same spectrum, then there should be naturally occurring mutations that can result in both. And as a matter of fact, that is the case. So there have been these large families that have been studied for a long time that have mixed ALS and frontotemporal dementia. So some people in the family have ALS, some other people have FTD, and very unfortunate few have combined ALS and FTD. This used to be called chromosome 9 linked ALS and FTD until about 12 years ago in 2011, when the gene for this was finally identified, and that's called C9 or F72, or chromosome 9 linked open reading frame 72, as I'll talk about later. But that's just a demonstration that the genetics, research into genetics has really exploded over the past 10 to 20 years. So back in 2004, almost 20 years ago, there were really only 3 genes that we knew of that were associated with the development of ALS and perhaps frontotemporal dementia. And these explained maybe 20% of familial ALS. And remember that familial component of ALS is only 10%, right? So these genes explain maybe 2%. And fast forward, to 2021, which is the last time I sort of cataloged all the mutations. There have been maybe one new one since then, but there's a lot. There's over 34 different mutations, genes that are mutated in both ALS and FTD. And so now we can explain 8 out of 10 the reason for disease onset, and 8 out of 10 people with familial disease, and even a good 17 to 20% of those with sporadic disease who have de novo mutations in these genes. So now we can actually order these and use this information to learn something about the pathophysiology of disease. So it turns out that if we sort of categorize these based on their known function, there are 2 main gene ontology groups that come out, and those include protein turnover, mechanisms involved in protein turnover, and RNA metabolism. This is the one reason that we believe this link between RNA and protein homeostasis is really critical for neurodegenerative diseases like ALS and FTD. There's also several genes that are involved in axonal transport and mitochondrial function that are likely to be related to both protein turnover and RNA metabolism. Now, when we think of which mutations cause ALS or mixed ALS and FTD, it's about 50-50. I would say a few more of these cause mixed condition than solely ALS. There's a couple actually that cause primarily FTD and minimally ALS, and I think this is a bit of a slippery slope. So some of us, including myself, believe that if we lived long enough, these mutations would probably people with these mutations would probably develop the other clinical syndrome. The problem is that ALS, because it affects the muscles that control breathing or eating, people usually don't live past a few years, and so there's a competing effect, and the pathology doesn't spread beyond those systems. And a similar argument can be made for FTD as well. All right, but what about the molecular pathology underlying these? So the large majority, about 2/3 of these genes result in the accumulation of TDP43, like we discussed before. And there are some outliers here, like SOD1, which happens to be the one that is studied most often in relation to the medications that were tested in clinical trials, because this was first discovered back in the 1980s. But in general, most of these cause TDP pathology. So that really suggests that that this is not like a one-off or a secondary change, but that it's an integral feature of many diverse genetic origins of disease. And as a matter of fact, mutations in TDP itself cause disease, right? So this again suggests that this is a critical feature, and that maybe this is a central node that all these mutations impact, and then all other downstream changes following TDP pathology and mislocalization are responsible for neurodegeneration. That's one possible interpretation of this. All right, so how common is this pathology? So we think about ALS, like I mentioned, 94-95% of people have TDP pathology, and particularly sporadic. And in familial disease, about 70%. Here's where SOD1 really stands out. And then in frontotemporal dementia, it's not quite as clear. So TDP is about 50% of the total, and tau is the other big contributor. And it's about the same between sporadic and familial disease. All right, just to summarize this part, ALS and FTD are closely related on the clinical side, definitely genetic side, and the pathologic aspects as well. All right, so let's talk a little bit about pathophysiology. What is this protein TDP, and what is it actually doing? All right, so we think about a healthy spinal motor neuron. These are large cells in the anterior horn. TDP, as I mentioned, is primarily a nuclear protein. It's involved in many aspects of RNA metabolism, primarily RNA splicing, but also transport, translation, and the stability of RNA. And in disease, though, it is fully excluded from the nucleus and instead accumulates within the cytoplasm of neurons. And we think that this is representative of a loss of function, because TDP can't do its normal job splicing RNA if it's not in the nucleus, and also a potential gain of function when it accumulates in the cytoplasm. So this is a cartoon of TDP. It has a nuclear localization signal that puts it in the nucleus. It has, previously there was an annotated nuclear export signal, but it turns out that's actually not functional. We know that TDP moves in and out of the nucleus. We're not entirely sure about the mechanism of nuclear export. Right now, I believe most evidence suggests that it's a passive transport. When it binds to RNA and gets sucked out of the nucleus as the RNA is transported out. It has two RNA recognition motifs that allows it to bind RNA. It has this really interesting low complexity domain within the C-terminus that has many names: glycine-rich, prion-like domain, unstructured domain. We believe that this is highly flexible, allows the protein to participate in many dynamic changes in its structure. So I mentioned before that mutations in TDP are associated with ALS and FTD, and now we know of almost 50 different mutations across the gene, most of which are centered in this low complexity domain. And of course, this implies that this domain is really essential for the function of TDP, and that these mutations can impair that function. So what is that function? As I mentioned, the best characterized role of TDP is in splicing. And that's because when you knock out or when you lose TDP43, you end up with the inclusion of regions of the genome that really shouldn't be in your RNA, and we call these cryptic exons. So this is an example of sequencing data showing this particular gene, and most of the reads come from exons, right? And then there's these little areas these introns get spliced out, and so they have low read counts. But when you don't have TDP, what happens is you get a new exon that wasn't there before. And so these go by the catchy name of cryptic exons. Now, on the face of it, this may not seem like a big problem, but when you have a new exon that's stuck in the middle of your coding frame, it usually causes a shift in the reading frame. So instead of having that regular 3 nucleotide pattern, if you have any other number that's not a multiple of 3, you shift the reading frame, you end up with a premature termination codon, which leads to nonsense-mediated RNA decay of that transcript and a loss of function. And when you take into account that TDP recognizes binds to over a third of all transcribed genes, when TDP is not around in the nucleus, this can lead to a lot of problems, right? And one way to think about it is it's death from 1,000 cuts, right? All these things are going to be affected by the loss of TDP. But the question has arisen, are there one or more things that are primarily affected that when they're lost can lead to neurodegeneration? And there's just a couple that I want to go over really quickly because these are going to turn into the basis for new therapies. So one of the most common is Stathmin-2. And here, these are data from ALS patients, right? So we're looking at the splicing of this gene, Stathmin-2. And again, these peaks, even these small ones, correspond to exons. And this is the pattern in otherwise normal people. And this is what happens in ALS. You have this new peak, which corresponds to a cryptic exon. You see this in almost 100% of people with ALS. It's really incredible, very consistent. It maps with the TDP43 binding site in that intron. And what happens is that instead of getting this full-length gene product, you end up with a truncated one that is highly unstable, and you don't get a Stathmin-2 protein. So as I mentioned, this is really consistent. So people have started to use this as a biomarker of disease. And so you can actually measure that truncated or abnormal Stathmin-2 transcript in people with ALS that have TDP pathology versus SOD1. And remember, I said that this is a big contributor to familial disease, was studied a lot before, but we think it's different than the majority of people with ALS, and this is very consistent with that interpretation. It turns out that in FTD, in people with a TDP pathology in FTD, which we call frontotemporal lobar dementia with TDP pathology, they also show Stathmin-2 misprocessing, and you don't see that in these other pathological subtypes of FTD, so it's very specific. It's even found only in the tissue types that are affected in disease. And so, again, in ALS, you find the majority of it in the motor cortex and in the spinal cord, but not, say, in the cerebellum, the occipital cortex, or even the hippocampus. In frontotemporal dementia, again, you find this in the frontal and temporal cortex, but not in the cerebellum, right? So this is a really useful means of distinguishing people that have TDP pathology. And as a matter of fact, this is now being developed as at least we're trying to develop this as a means of stratifying people before they go into clinical trials to say whether or not they'd be candidates for therapy targeting TDP43. Now, on the other side of it, is there any therapeutic potential to replacing Stathmin? And that's the basis for, there's at least 2 companies that are developing approaches to restoring Stathmin-2 expression, and one of them is Curalis. I think the jury's still out on whether or not replacing this one gene will have any impact on disease that's characterized by missplicing and loss of so many different gene products. But I think, actually some of the data from the mouse models suggests that this is essential for motor neuron survival. So we will see. And that's the good thing, is that we're actually going to learn more by doing these trials. All right, I'm going to switch gears a little bit and talk about now the single most common genetic mutation responsible for both ALS and FTD, and those are the mutations in this gene that I mentioned before, C9ORF72 or chromosome 9 open reading frame 72. So when we look at the genetics of ALS and FTD, mutations in C9orf72 are really the largest contributor, right? So there's a few mutations in FUS or TDP or SOD1, but the large majority come from C9orf72, and together we can explain almost 70% of disease, at least familial disease, and 11% of sporadic. And this is a little outdated. I think the numbers are more like 75% familial and almost 20% of sporadic disease based on these mutations. Again, it comes down to how hard do you look and how many genes do you include in your panel. So this is a map of the C9orf72 gene. The most important thing to realize is that this red spot here between two introns is where the repeat is actually located. The mutation, this is a region that is rich in G's and C's. It's actually a repeat of GGGGCC. And people who are otherwise normal have about 6 to 8 repeats usually. But in ALS and FTD, the number of repeats can expand up into hundreds or thousands. And what the actual threshold is for disease, we're not sure. A very conservative estimate is greater than 23. I think it's probably more in the 30s. But you can imagine, I don't know if anyone is familiar with sequencing technology. Shotgun sequencing, what we do is we break the genome into small pieces, sequence them, and then try to reassemble them. If you just have a bunch of G's and C's, you're never going to be able to reassemble it. And that's why this mutation was missed for so many years until people developed specialized techniques like repeat prime PCR to detect specifically repeats. All right, so what happens when you have this huge repeat in the leading region, the intron of this gene? So one thing is to get you reduced expression of the RNA, and you can see that when you have the repeat, you have reduced expression of some of the transcript isoforms from this gene. We think that the protein, C9orf72 protein, is required for proper autophagy, so protein turnover. And when you don't have that protein, when you don't have functional autophagy, you can have the accumulation of proteins like TDP43. There's also some evidence that the repeat itself in the RNA can cause problems, and it does that. It can do so by sequestering RNA binding protein. So if you think about a protein that binds GGGGCC, if you have 8 repeats, then maybe you have 8 proteins stuck to it. But if you have thousands of repeats, then you have thousands of proteins that are stuck to it, and you can actually deplete that protein from the rest of the cell and impair its function. And so we can see that using fluorescence in situ hybridization, or FISH. You can see these big foci that are rich in the G4C2 sequence in cells that carry the repeat. Now, the other really interesting thing is that this repeat, even though it's not in the coding region of the C9orf72 RNA, it gets translated. Although there's no canonical start codon, there are things that look sort of like a start codon, enough for the translation machinery to get started and make protein. But because this is a repeat, you end up with a repetitive sequence of peptides. And it's, conveniently enough, it's 6 nucleotides. So you end up with a repeating sequence of 2 peptides. In this case, glycine and alanine. But again, there's not a true start codon, so there's not a true reading frame. So as this gets shifted, you can get translation in any reading frame. You can also get translation in the antisense reading frame. So you end up with several different dipeptide repeat proteins, many of which can be detected in people with C9orf72 ALS FTD using specific antibodies against these dipeptide repeat proteins, including glycine, alanine, glycine proline, and glycine arginine. And again, people have taken advantage of this and now use these dipeptide repeat proteins as biomarkers of C9orf72 related ALS FTD. There's also many potential strategies that are being designed to prevent the translation of these dipeptide repeat proteins, assuming that they are in fact toxic and part of the pathophysiology of disease. So just to review, I won't go into many of the details, but these GGGGCC or hexanucleotide repeats can have several effects. They can impair the translation of the protein from this gene, resulting in a reduction in the C9orf72 protein and impairment in autophagy and protein turnover. They can also have gain of function effects by sequestering RNA binding proteins that have many essential functions, and they can generate these dipeptide repeats or DPRs that have many pleiotropic effects on cellular function, right? So, this provides many opportunities, both in terms of biomarkers and treatment. So, there's now gene therapy to replace C9orf72 protein and ASOs antisense oligonucleotides that can anneal to this a particular part of the RNA and prevent it from causing dominant negative effects or impairing translation of the C9orf72 protein. People are using, so these RNA foci tend to be very difficult to detect, and so many people are instead focusing on dipeptide repeats. These are now great biomarkers for disease. So there's ELISAs and even single molecule technology that's available for detecting dipeptide repeat proteins in cerebrospinal fluid and in brain. And they're being used not just as a marker of the C9orf72 mutation, but also as target engagement biomarkers. So if you deliver a therapy that acts on this gene, you can actually see the dipeptide repeat proteins disappear from the CSF of treated individuals. These tests have been done not just in animals, but also are beginning to be done in people. And again, one of the approaches is to use antisense oligonucleotides to prevent the translation of these repeats into dipeptide repeats. And this is also being approached using small molecules. It's possible that by targeting only a single aspect, like the DPRs or the C9orf72 function, that that's going to be insufficient to prevent disease. But and as a matter of fact, some of the recent trials suggest that may be the case. But again, we are learning more as we conduct these studies. All right, just to summarize, so I've provided some evidence that suggests that ALS and FTD are closely related to one another. There's clinical, neuroanatomical, and genetic and pathologic overlap. The pathology is in many cases characterized by the accumulation of TDP43. Almost all people with ALS and about half of people with frontotemporal dementia. And the single most common mutation responsible for ALS and FTD is in the same gene, and that is C9orf72. There are many biomarkers that are now being developed that can tell us what is going on a molecular level in disease. One of the ones that we talked about was Stathmin-2 missplicing. I didn't talk about it, but a second misspliced gene that turns out to be also very consistently misregulated in ALS and frontotemporal dementia with TDP deposits is UNC13A. And this is gathering a lot of excitement lately because it turns out that UNC13A was also the only gene identified by genome-wide association studies as being linked to risk of developing ALS. So now we have not just genetic evidence of TDP-related missplicing in ALS, but this suggests that if you could somehow correct UNC13A missplicing, you can affect the disease course or the risk for developing disease. So that is a area that many people are focusing on right now. And finally, we talked about C9orf72 and how expansion mutations in this gene can have a multi-pronged effect on autophagy via the loss of the C9orf72 protein, sequestration of RNA binding proteins, and also the production of potentially toxic dipeptide repeat proteins. And these are being developed. These different disease mechanisms are forming the basis of biomarker and therapeutic target design. All right, and just to follow up with the references here, there are many fantastic references and resources that go over some of the material that I've talked about today. I would be more than happy to direct people to additional sources of information if desired. Thank you.